morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Custom Library Adapters via Enzymatic DNA Synthesis Improve Precision of Real-Time Nanopore Sequencing. This webinar is sponsored by DNAScript. Our speakers today are Dr. Tim Mercer, Associate Professor at the Australian Institute of Bioengineering and Nanotechnology at the University of Queensland, and Bailey Schmidt, Senior Marketing Manager at DNAScript. You may type in a question at any time during the webinar, and you can do this through the Q&A panel, which appears on the right side of the webinar presentation. We will ask our speakers your questions after the presentations have concluded. If you look at the bottom tray of your window, you will also find a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Dr. Mercer. Please go ahead with your presentation.
So thanks for joining me today. My name is Tim Mercer from the University of Queensland, and today I wanted to present some of our work on the applications of enzymatic DNA synthesis. But before I begin, I want to thank GenomeWeb for hosting us, as well as for DNAScript for sponsoring this presentation, as well as yourselves for registering and tuning in. So today I want to talk about the applications of enzymatic DNA synthesis, and specifically how we integrated reference controls into library adapters to try and improve next generation sequencing. And the way I want to do this is, firstly, I want to describe some of the foremost applications of the new enzymatic DNA synthesis capabilities of the DNAScript technology. I then want to describe how we plan on integrating these on-demand synthesis capabilities into our new mRNA manufacture facility being established at the University of Queensland. And then finally, in the second half of the talk, I want to describe how we've used enzymatic DNA synthesis to build control library adapters that improve the qualitative and quantitative accuracy of Oxford Nanopore sequencing. So let's start with a quick introduction about enzymatic DNA synthesis. So enzymatic DNA synthesis is a relatively new technology that uses enzymatic rather than phosphoramidite synthesis to build DNA sequences. So enzymatic DNA synthesis typically uses the terminal deoxynucleotidyl transferase enzyme, or TDT enzyme. This is the enzyme that's responsible for adding extra nucleotides to the gene sequences encoding the T and B cell receptor sequences. And this is done during a process called somatic hypermutation that's used to expand antibody diversity. However, since its discovery in the 1960s, researchers have quickly realized the potential of terminal transferase to perform controlled enzymatic DNA synthesis. However, it's really only been recently that this potential has started to be realized. Recently, DNAScript developed a benchtop instrument called the Syntax that can enzymatically synthesize DNA oligonucleotides in under a few hours. And why is this enzymatic DNA synthesis more beneficial than the conventional alternative phosphoramidite approach? Well, firstly, it doesn't require the toxic and volatile chemicals that are needed for phosphoramidite synthesis. Secondly, enzymatic synthesis is more accurate. It can achieve a 99.7% accuracy compared to around 98.3% for phosphoramidite synthesis. And finally, and this is what I think is the most compelling advantage, is the potential for enzymatic synthesis to routinely synthesize long sequences. And this is something which I want to return to shortly. But first, let me quickly run through some of the current applications that use the DNAScript Syntax enzymatic synthesis instrument. Oligonucleotides printed using the Syntax instrument have been applied for many very well-established applications. So you're probably well aware things like primers for PCR and qPCR, primers for amplicon sequencing, and libraries for mutagenesis as well. And in each of these cases, the major advantage is that a user can quickly print primers or oligonucleotides on demand, so custom primers according to their specifications, and within a few hours for these particular applications. In addition to these established applications, there's a range of newer emerging applications on the horizon that are currently being developed and verified. And later in the talk, I hope to add another example to these use cases, and that's the example of printing NGS library adapters, custom NGS library adapters. However, it's this last one here, the gene assembly, where I actually want to focus for the moment. So let me just quickly touch on, the use of oligonucleotides, printed oligonucleotides for gene panels, fluorescent hybridizations, for hydrolysis probes, but also this one here, this gene assembly. Now, gene synthesis is currently achieved by stitching together many small oligonucleotides into a larger synthetic gene, or even a genome. Now, this is the same process that's used for phosphoramidite synthesis. However, for phosphoramidite synthesis, the efficiency of the synthesis steadily drops off beyond about 200 nucleotides, and this requires these larger scale assemblies to be gradually constructed in stages, in sequential stages from these smaller strands. However, this limitation, this 200 nucleotide limitation, does not necessarily hold for enzymatic synthesis. And enzymatic synthesis has the potential to generate long sequences that are otherwise out of reach using standard chemical synthesis. And while I must highlight that this potential has not yet been realized, it's our eventual aim to use enzymatic synthesis to print entire genes and genomes in a single run. And for me at least, it's really this potential that most excites me about the enzymatic DNA synthesis technology of the Syntax instrument. And the reason for this is because given the emergence of RNA as a sort of an exciting new therapeutic modality, and this has been most notably demonstrated by the successes of the Pfizer and Moderna vaccines, we recently established a joint facility at the University of Queensland to manufacture RNA therapies, or mRNA. And the aim of this facility is to build mRNA vaccines and RNA therapies for academic and industry partners and to drive new research in this burgeoning field. And in this case, the first step in our manufacture workflow is the synthesis of DNA sequences of interest. So here we plan to incorporate the DNAScript syntax instrument to synthesize the sequences on demand. This then forms, these synthesized sequences then form the DNA template that's used for downstream in vitro transcription reactions that are ultimately followed by purification and analytical verification. Now, given this ability to rapidly synthesize and assemble the sequences, combined with a relatively quick RNA manufacture workflow, we aim to achieve end-to-end manufacture, so from initial design to the final purified mRNA product within 24 hours. And this, if we can achieve this, this can essentially enable extremely rapid and personalized manufacture of RNA therapies on demand. So that provides a quick introduction into the enzymatic DNA synthesis technology and how we plan to use it to develop new and innovative RNA therapies and technologies. But for the second part of the talk, I actually want to describe some of the new and innovative applications that we have already completed. In this case, we use the syntax to print custom library adapters for Oxford Nanopore sequencing. And I like this idea that we're essentially teaming up enzymatic DNA synthesis with enzymatic DNA sequencing. And so together, these two processes, these two enzymatic processes, can enable very rapid design, build, and test iteration cycles, and really allow us to quickly explore new and novel and innovative RNA technologies and designs. So, look, let me first start by describing what a library adapter is. Library adapters are short oligonucleotides that are ligated onto the ends of your sample DNA during library preparation. These adapters include sequences such as primer binding sites that are needed for the sequencing reaction. Over the years, additional functional elements have also been added to these library adapters. So for example, many adapters now include unique molecular identifiers that can be used to identify reduplication that occurs during PCR amplification. Alternatively, they may also include sample indexes and barcodes that allow multiple samples to be multiplexed together in a single sequencing run. Or alternatively, they can be used to identify, for example, single cells, specific single cell combinations. And what we wanted to do was we wanted to add a new functional element to library adapters that we term the control sequence. So this new functional element acts similar to a reference standard or a reference control in that it provides a measure of diagnostic performance. We call these adapters that contain these control sequences, we call them CAPTORs, and this is short for control library adapters. And in the case of Oxford Nanopore sequencing, at least, in addition to acting as a bit of a reference standard, it can actually also allow us to interface with the surrounding bioinformatic architecture and dynamically control library sequencing as well. Okay, so let's just put a pin in that idea for the moment, because before I describe how the CAPTORs work, let me quickly take one step back and describe how nanopore sequencing works. Now, nanopore sequencing occurs by measuring the electrical current across the pore that occurs as a DNA molecule is fed through the nanopore. And as the DNA passes through the nanopore, the changes to the electrical current can be used to determine the sequence in real time. Now, for the purposes of our discussion, one of the important things to remember is that the library adapter is actually the first part of the DNA that feeds through the nanopore. So it's the first part of the DNA that's sequenced and read by the nanopore. Now let me return to our Captor design. So as mentioned, the CAPTORs are like any normal library adapter, except they have this additional control element that's shown here in red. Now the control element comprises a single variable sequence that's flanked by these two constant sequences. And this variable sequence is actually different between different CAPTORs. So using the DNAScript syntax instrument, what we did is we actually synthesized a pool of 72 different CAPTORs, and each CAPTOR had a different variable sequence. Now the variable sequence was about 30 nucleotides, and together all the variable sequences from the different CAPTORs essentially encompassed all the different sigmas that are used by Oxford Nanopore sequencing for their base calling. So in this case, all the sigmas are present at least once in the CAPTORs. And together, when you combine all the 72 CAPTORs together, you encompass all of the possible sigmas. Now to demonstrate, to provide sort of a proof of principle about how the CAPTORs work, we performed a metagenome experiment. In this case, we collected samples from the Sun Lake in the crater of the Nevada Toluca volcano. So it's about a day's hike outside of Mexico City. Don't worry, it's not very active, but as you can see, it is pretty stunning and beautiful. We then took those DNA samples and we extracted the DNA from them, and we used them to prepare libraries with the CAPTORs. So here this schematic shows the library preparation and sequencing workflow. And I think this is really one of the greatest advantages of the CAPTORs, because unlike other reference standards, CAPTORs don't require any additional steps, but they're really seamlessly integrated within the library preparation workflow. So in contrast to other reference standards and controls, such as spiking controls that need to be added to a sample, or reference materials that need to be sequenced in their own library, the CAPTORs don't require any additional steps or any additional fiddling around. In this case, once prepared, the library sequenced as normal, in this case using a MinION instrument. The CAPTOR sequences are then retrieved from the end of the reads and analysed. And the analysis of those CAPTORs really starts to provide quantitative and qualitative measurements of the library performance. And given that this sequencing data is generated in real time for the Oxford Nanopore sequencing, we can also analyze the CAPTORs in real time. And this provides an ongoing measure of sequencing performance across the duration of the experiment. So what does this analysis of the Oxford Nanopore, of the CAPTORs look like? And what does it tell you about your particular library? Now, the first thing we measured using the CAPTORs was sequencing accuracy. In this case, we evaluated the accuracy by which each sigma within the variable regions has been determined and called. And this provides essentially a measure of accuracy for the ONT-based calling. So here's a few examples here from an example variable sequence. You can see the different sigmas and the different sequencing accuracy associated with it, with each of the individual measurements generated by different instances of the sigmas across each of the 72 CAPTORs. And you can kind of easily see how we can use this to evaluate our sequencing accuracy and build quite a detailed error signature for each library. And it's quite comprehensive as well because it essentially incorporates every sigma. So here this graph plots the sequencing error rate for each KMA that's been ranked along the x-axis. And this graph, it really shows you both the distribution of sequencing accuracy for the different sigmas. So you can see that distribution, the range in sequencing accuracy profiles. But you can also see the variability between the different replicate libraries as well. So this allows you to see the variation in sequencing library accuracy between different libraries. And here the CAPTORs are really acting as internal reference controls, much like spiking controls, to measure the sequencing performance of each individual library. Similarly, in this case, we can also use the CAPTORs to benchmark the accuracy and the performance of different reagents, different instruments, different protocols, different methods, even different users. Here what we've done is we've actually benchmarked the accuracy of the standard R9.5 Nanopore, which is slightly worse off than the newer 10.3 Nanopore version, which has a longer barrel and therefore can more accurately resolve longer homopolymer stretches that have been a challenge for nanopore sequencing. And again, this shows you how CAPTORs can act essentially like an integrated reference control that evaluates the sequencing performance of that particular library. Now, given that the CAPTORs are attached to each read, they can also be used to measure the sequencing accuracy at a per read and a per pore level. So this is a schematic figure of the flow cell, and each circle represents a nanopore well with the color reflecting the mean performance across time. And in those sort of inserts along the bottom and side, you can see the performance of each individual read shown as a dot, and in each individual pore across the duration of the sequencing experiment. And this really demonstrates the granular resolution at which CAPTORs evaluate sequencing performance and provide unprecedented information at the read, the pore, and the flow cell level. Now, why would such high-resolution data be useful? Well, another sort of exciting feature of nanopore sequencing is called adaptive sequencing, or read-until sequencing. In adaptive sequencing, the nanopore reads the initial part of the DNA molecule, and if it's not interesting, it reverses the polarity to spit the DNA sequence back out. And this is great because you can enrich for sequences of interest while excluding unwanted sequences. Now, because our CAPTORs are the first sequences that pass through the nanopore, they can provide an initial indication as to whether a read is good quality and whether the nanopore should continue to sequence it or not. And if the CAPTOR, which is the first sequence, provides an initial indication of poor quality or a duplicated read, the nanopore can prevent it from being sequenced. And the benefit is that we can integrate this per read data provided by the CAPTORs with adaptive sequencing to provide dynamic control of nanopore sequencing. Now, in these previous slides, we've shown how CAPTORs can provide a quantitative evaluation of sequencing accuracy and performance. But they can also provide a quantitative evaluation of sequencing performance. what they can do is they can measure the quantitative accuracy and the quantitative complexity of a library. They can sort of, I guess, evaluate that quantitative dimension during the sequencing experiment. And the way they can achieve this is actually quite simple. In this case, what we do is we combine the captors at different concentrations. So they sort of form a serial dilution or a staggered concentration within a master mix. And this means that within that master mix, some captors are rare, while other captors are abundant. This staggered master mix is then used to prepare a library as previous. So in this case, we've used the captors in an RNA sequencing experiment. We've used the captors to prepare a library from a reference RNA sample that's then sequenced on the MinION. And as you sequence your library, what you do is you can compare the count of captors with their known concentrations in the master mix. So on the left-hand side, you can see the plot. And what it does is when you compare the observed count of the captors, so the observed count of the captors as they come through versus their expected concentration in the master mix, when you plot this, it forms this nice staggered quantitative reference ladder that indicates the quantitative performance of the library. Furthermore, because the captors are ligated to the termini of the DNA fragments at a constant ratio, it actually ensures that their quantitative counts and their dynamic range are in direct proportion to the quantitative profile of the accompanying sample. So in this case, the captors are ligated 2 captors per read, which means they're in a constant ratio in proportion to the gene expression of genes in the accompanying RNA sample. And as a result, the captors can actually provide a direct measure for the quantitative accuracy and the complexity of the library. And you can see this here. You can see that by using that staggered ladder, you can evaluate the sensitivity and the limit of detection for a library. You can also evaluate how accurately we measure gene expression in the sample, and you can actually empirically measure the variation, the quantitative variation that's apparent in our sample and that's apparent at different sequencing depths and different abundances as well. Now, given that sequencing is also occurring in real time, we can use the captors to measure the quantitative profile of the library in real time as sequence is being performed. So what we can do is we can evaluate its quantitative performance, and once we reach a predetermined threshold of sensitivity, the sample can be stopped from sequencing. So this can be used, you can envisage it being used to ensure that if you're running, say, a particular clinical diagnostic sample, that you ensure that you achieve sufficient sensitivity and limit of detection. The other thing which is key is we can actually use this linear regression, shown here as a dashed line, to directly convert between the read counts, the observed read counts of the captors, and their known molar concentrations. And what this allows us is to actually move from subjective read counts, so reads per million that's typically used in next generation sequencing, to a much more objective unit of measurement, such as moles, molar concentration. And this is important because this actually starts to allow us, once we start to move to those objective quantitative standard units, it allows us to start directly comparing abundances between samples. Because the captors provide this empirical standard unit, they provide a universal quantitative reference that we can use for comparing and normalizing between 2 samples. So that is that any 2 libraries that have been prepared with the captors can actually be accurately normalized using the reference captors to remove unwanted batch effects and differences. And to demonstrate this, what we've actually done is we've run on our samples a range of different normalization methods. You can see here on the left, these are actually the different replicate samples that have undergone no normalization, and the reference log expression shows really the distribution of reads in each individual sample. In the middle, this is kind of the current best practice techniques for normalization. Things like trimmed medians, things used, for example, normalization methods used, for example, in EdgeR and DSEC. And on the right-hand side, you can see that what we've done is we've used the IUVG normalization method, and which is typically, this is an algorithm that's typically used to normalize between samples based on internal spiking controls, but here we've trained it on the actual CAPTORs themselves. And in this case, what we've actually done is we've normalized using the CAPTORs, and you can see how that's essentially improved our normalization between samples. And in this way, we can actually use the CAPTORs to normalize between samples, remove technical variation, and enable more accurate comparisons between samples. And indeed, when we look at the accompanying sample, the accompanying RNA sample, when we use the IUVG with our CAPTORs, we can actually more accurately and reliably detect differences in gene expression between samples using the CAPTORs. We can also use, as I mentioned before, because the CAPTORs provide this empirical measure of technical variation that's aligned or assigned according to the quantitative abundance, we can actually use that estimate of technical variation to determine whether observed fold differences between samples are actually significant as well. So this is quite different to the current bioinformatic methods that are used to identify biological differences between samples. Now, one of the benefits of this is we can actually extend this to beyond comparing samples within a single experiment, but instead use the CAPTORs to compare any experiment, compare any samples between any experiment from any laboratory that uses the same CAPTOR master mix. So in this case, any libraries that are prepared using the same CAPTOR master mix can be compared regardless of how, when, or where they were sequenced. And this is particularly important for point-of-care sequencing approaches, such as using the handheld MinION device, that can be often performed under different heterogeneous conditions and can suffer from these batch effects. Unfortunately, we estimate that our pool of CAPTORs that we've synthesized using our DNA syntax instrument is sufficient to prepare millions of libraries. So we can actually use these pools of CAPTORs to build large networks of samples that can be accurately compared. And this is going to be particularly useful for the analysis of patient samples that are often confounded by technical variation. So this is large patient cohorts or across longitudinal patient timelines or between different clinical laboratories. This sort of interoperability, this sort of ability to compare between samples will really be key in integrating and comparing the expanding number of samples that will be sequenced using different methods, protocols, and in different laboratories over the next few decades. So let me sort of end on that forward-looking note. You know, this idea that we can use CAPTORs to start to build these large networked databases by which to compare samples. And let me quickly summarize essentially what we've found and what we've achieved with this enzymatic DNA synthesis. I mean, enzymatic DNA synthesis can enable on-demand rapid DNA synthesis. At the University of Queensland, we intend to harness this opportunity, these advantages, to develop novel, rapid, and personalized RNA technologies. Reference controls as well are a central requirement that are required to ensure the accuracy and reliability of next-generation sequencing for clinical diagnosis. And the incorporation of reference controls within our library adapters, as we've achieved here with the CAPTORs, ensure that these benefits can be seamlessly integrated within NGS libraries without requiring different additional steps. Now, in this case, we use the syntax instrument to design and build CAPTORs, these custom library adapters for Oxford Nanopore sequencing. However, we could also equivalently synthesize CAPTORs for other sequencing platforms, such as short-read Illumina sequencing. Furthermore, really any technology that uses barcoded adapters, such as single-cell sequencing and spatial transomic sequencing methods, can similarly incorporate these quantitative reference control sequences and confer many of the benefits of CAPTORs on these methods. So you can easily envisage using CAPTORs to measure the complexity of single-cell libraries, and use CAPTORS to inform normalization between the transcriptomes of individual cells, for example. More broadly, taking a little bit of a step back as well, we really believe that the rapid on-demand enzymatic DNA synthesis that's achieved by that benchtop DNAScript syntax instrument really provides us with a really wide space, a really wide informational space that we can explore, and we can take that information and we can essentially encode it within NGS libraries. So what we can do here is we can, on demand, we can design custom spike-in controls or custom adapters, custom barcode adapters, and we can essentially use that as a way of building and designing information that is then hard encoded within our libraries. And this information, which is encoded within the libraries, can then be later retrieved and analyzed by a responsive bioinformatic architecture. And for me, that's one of the most sort of exciting things about this on-demand DNA synthesis technology. It's this ability, this flexibility, where really it's just your imagination and creativity that limits the types and the forms of information that can now be easily encoded within next-generation sequencing libraries and then can be later retrieved and interface with that surrounding bioinformatic architecture. So on that point, I would like to finish up. But before I do, I want to thank a lot of the hardworking scientists who have helped with this work today. This includes Helen and Scott, who have performed much of the analysis and interpretation of the CAPTORS, as well as Andre and Bindu and Ted and Ira, who helped design the CAPTORS and perform the laboratory experiments. I also want to thank the team, Jeff and Marike, who have helped support this work. I hope they let us continue to build new and cool and funky ways to build new technologies using their enzymatic DNA synthesis technology. I'd also like to thank David Barter at the Garvan Institute for his support in these projects. And on that note, I want to finally finish off by handing over to Bailey from DNAScript, who's going to follow up from this talk to provide a bit more background on their work at DNAScript and some of the exciting developments that are going on there. So thank you very much.
Thank you, Dr. Mercer, for that great presentation. To wrap up this webinar today, I would like to provide an overview of DNAScript and our syntax on-demand synthesis system. Founded in March of 2014, DNAScript has been focused on pioneering the development of enzymatic DNA synthesis, also known as EDS technology. In September of 2015, we synthesized our first enzymatic codon, and ever since then, we have continuously worked to increase our oligo length capabilities, with our first 10 nucleotide oligo synthesized in December of 2016, first 50 nucleotide oligo synthesized in September of 2017, first 150 nucleotide oligo in September of 2018, and 200 nucleotide oligo in November of the same year. At the time we were able to synthesize an oligo of 200 nucleotides, we initiated the development of a benchtop instrument and transitioned from EDS on a lab bench to EDS in an automated system that can produce 96 6-tumor oligos in a single run. Since then, we have successfully reached the commercial launch of the syntax system on June of 2021, with the aim of powering the biorevolution with on-demand DNA. The syntax system is the first product of the syntax platform leveraging enzymatic DNA synthesis technology and is available now. The complete syntax platform is truly an end-to-end solution and is composed of benchtop instruments, reagent and consumable kits, and a suite of software. This slide displays the current capabilities offered with the syntax system. We're able to synthesize up to 96 oligos in parallel with 200 to 300 picomole per well. It requires less than 15 minutes of hands-on time. You can get 20 mers in 6 hours or 60 mers in 13 hours. The syntax supports 15 to 60 nucleotide de novo synthesis, and you can have customizable iDNA of 15 to 40 nucleotides. We use 4 inks of A, T, G, and C, and this is a fully automated system where you simply put your sequences in and get oligos out. Our vision is to power a biorevolution by democratizing DNA right, making DNA synthesis as accessible, flexible, and on par with DNA read and DNA edit. Very much like sequencing and CRISPR genome editing are accessible life science tools today. The Syntax system enables enzymatic DNA synthesis on the benchtop. The combination of an enzymatic approach and a benchtop form factor offers several advantages over ordering oligos from service providers using traditional chemical synthesis methods. First, the Syntax is powered by EDS, an aqueous-based synthesis approach that we continue to develop and improve as our platform evolves. In contrast, traditional DNA as a service is based on chemical synthesis, an approach where methods have not significantly improved over time and uses large volumes of harsh chemicals. The Syntax solution is a simple integrated experience which is accessible to any lab as compared to DNA as a service vendors where transactions often involve multiple touch points and supply logistics may be complex. Additionally, turnaround times with service vendors may be inconsistent and unreliable, while Syntax allows dependability as oligos are produced on demand in the lab when needed. This also provides greatly increased speed for same-day results from start to finish. Lastly, Syntax on-demand oligos provide ultimate control as the user defines which oligos are printed and when they're printed. This combination of speed, control, and dependability supports the acceleration of the iterative design, print, test cycle ever present in molecular biology and genomics applications. The ability to print 20 to 30 mer oligos in 6 hours enables you to print a set of oligos, test them in your downstream application that same day, then design and print new oligos based on your results for immediate use the next day. So again, the Syntax system is available today. You can find more information by visiting our website, DNAScript.com. If you have any questions, please feel free to reach out to us at contact@DNAScript.com. We look forward to hearing from you.